Good morning, students. Today we are going to talk about a very important topic, that is the search of genetic material. So now the most important question, which molecule is actual genetic material? So till now we discussed about DNA in detail. So we got to know how exactly double-stranded structure of DNA is formed, how it was discovered. We know all that. We know about complementary base pairing technique. Now the question is, how was it proved that DNA is actual genetic material? Because before, nobody knew that which molecule is actual genetic material. They just knew from whatever was discovered before, like first experiment performed by Gregor John Mendel, and Mendel gave a term factor, which later came to know as gene. So as per Mendel, factors were one which carried genetic material from one generation to next generation, as per Mendel. Mendel gave the concept of factors actually. And after Mendel, many other scientists came. One of them was Thomas Morgan. So Thomas Morgan gave the chromosomal theory of inheritance. So he spoke about genes. He also spoke about chromosomes. He said that the chromosomes play a very important role in the process of inheritance. So there is something in chromosomes which is carried from one generation to next generation. So that was told by Morgan. There were other scientists, Boveri and Sutton. They gave Boveri-Sutton theory, that stating that chromosomal theory of inheritance. So they also spoke about the chromosomal level, at the chromosomal level. So in fact, if you talk about Morgan, he not only spoke about chromosomal theory, he also spoke about many new concepts like linkage. He also spoke about recombination. So he spoke about all the important terms. He brought a lot of change to what was given by Mendelian theory of inheritance. So this Boveri-Sutton theory again gave chromosomal theory. So all that was known, the genetic material somewhere inside the cell, somewhere inside the nucleus, somewhere inside the chromosome. But which molecule in chromosome, that was not sure. So when did we get to know which molecule in chromosome act as the genetic material? So it was in the year of 1926 that the mechanism of genetics of inheritance reached at the molecular level. Scientists started performing experiment, understand which molecule is responsible for genetic inheritance. So what experiment is performed in 1926 which helped in determining the particular molecule? Actually children, the experiment is carried out in 1928. There was a scientist named Frederick Griffith who performed experiment which is now popularly known as Griffith Experiment. So let us see what did Griffith do in his experiment. So let us see what was done in the Griffith experiment. Griffith experiment is with Streptococcus pneumoniae, right? It is written in the pink color. It is actually a bacteria that causes the disease pneumonia. So this is a back variety of bacteria, right? He took those variety of bacteria, Streptococcus pneumonia, and the two strains of bacteria were used. And these two strains were S strain and R strain. Basically, there are two varieties of this bacteria which are available. One is X strain and other is R strain. So let us see what is this S strain and R strain. So children, in S strain, here S stands for smooth. यहाँ पे S का क्या मतलब है? Smooth. So those variety of bacteria with had smooth polysaccharide coat on their outer surface, they are said to be as S strain of bacteria. Understood? यहाँ पे S का मतलब है smooth surface, और ये smooth surface किसका बना होता है? पॉलीसेकेराइड का. तो ये बैक्टीरिया क्या कहलाए? ये वेराइटी ऑफ बैक्टीरिया क्या कहलाए? S strain. And bacteria had a coat which is smooth or slimy mucus coat, right? Now the other variety of bacteria is R strain. Now R is for rough. They did not have the polysaccharide coat, so that is why it is a rough outer surface, and it is the only reason for the rough appearance from outside. So they lack the coat, and therefore they are easily destroyed by the immune system of the host body. क्योंकि इसमें पॉलीसेकेराइड का बना हुआ कोट नहीं होता, इसलिए इस बैक्टीरिया के पास प्रोटेक्टिव कवरिंग नहीं होती. So that is why they are easily destroyed by the immune system of host body. Now children, due to the presence of polysaccharide coat, it is something very obvious, right? So let us suppose you are present in your house. I'm just giving you an example. You are present in your house and your house is like a coat for you, right? It is a covering for you, giving you more protection. So if somebody tries to attack you from outside, so it is difficult to attack on you because you are being protected in your house. So that is the scenario for S strain bacteria. तो यही हाल कुछ S strain bacteria का है, क्योंकि वो अपने पॉलीसेकेराइड के कोट में प्रोटेक्टिव होता है. But for the R strain bacteria, they do not have the coat. Now since the coat is not present, they are more vulnerable to attacks. They get easily destroyed by the immune system of host. So these bacteria, they often lives as parasite. They often live inside the body of host. So if the host organism want, they easily attack and destroy R strain bacteria. So these are the two strains. So one is with a polysaccharide coat, that is known as S strain, and the other one is without polysaccharide coat, which is known as R strain. Now Griffith used these two type of bacteria in his experiment. So let us see how these two bacteria uses two variety of same bacteria helped. Now as from this we get to know that the S strain is virulent. Virulent का मतलब, it means it cannot be easily destroyed by somebody else. तो ये virulent हो गई S strain, क्योंकि किसी से destroy नहीं कर सकता इसे कोई. It can actually resist. So S strain is virulent, more resistance, right? It could cause disease, क्योंकि इसे कोई ख़त्म नहीं कर सकता, तो ये क्या करेगा? Disease cause कर सकता है. So let us see what happens. Now in Griffith experiment, what he did, like children, in this picture it is easily visible that first he injected the S strain bacteria in a mice. सबसे पहले उन्होंने क्या किया? एक S strain bacteria को mice में inject किया. He took a mouse, which was living, obviously. Then he injected into the mouse a S strain of Streptococcus pneumoniae bacteria. And what happened? It was seen that mice died by pneumonia. Why? Yes, exactly. Because as I said that S strain is virulent. S strain is being virulent, it helped pneumonia develop inside the mouse. As a result, mice died in a few days. Now, in the second setup of experiment, he injected R strain of bacteria in a mice. Next set of experiment में उन्होंने क्या किया? Mice में R strain को inject किया. Now what happened? R strain is non-virulent. It does not cause pneumonia. So as a result, mouse healthy, mice alive, no sign of pneumonia. So these were two scenarios. Now what did this prove? This proved that S strain were virulent, but the R strain were not. Okay. Now what he did? In the third step, he injected heat killed S strain. Now what is this? अब उन्होंने क्या किया? Heat killed S strain. Now what did Griffith do is that Griffith killed the S strain bacteria by heating them. तो इसे कहते हैं heat killed S strain. He found that by heating the S strain, the bacteria was killed. Now what you guess? If the bacteria which was already died injected into the mice, will the bacteria able to cause pneumonia? No. Right. Because the bacteria is already dead. So when he injected heat killed S strain in mice, he observed mice was healthy, mice alive. As per expectation, mice was alive, obviously. Now the most interesting part of this bacteria is the fourth one. So in fourth part, what happens? Now children, what he did, he took a mixture of heat killed S strain and R strain. अब उन्होंने heat killed S strain और R strain का mixture लिया. ठीक है? So what do you think? What happens? Now heat killed S strain was already killed. Even S strain is virulent but already killed. Obviously this is not cause pneumonia. What about R strain bacteria? R strain bacteria, we already saw that it is non-virulent. So this is R anyways non-virulent. So in this case mice should be alive. यही हम expect करते हैं. But what did he observe? He observed that the mice dies in few days, in which the mixture is injected that causes pneumonia in mice. So as a result, mice died. Now what Griffith concluded? Griffith concluded that there is something interesting which has happened in the mice. So this proved the fourth set of experiment, that something is being transferred from the heat killed S strain to R strain. कुछ ना कुछ ऐसा था जो S strain से R strain में transform हुआ. And due to that, now why it is S strain is virulent and R strain is non-virulent? S strain is virulent only due to the presence of polysaccharide code. हमने discuss किया. R strain is non-virulent because it did not have any polysaccharide code. That thing we have discussed. So what was the only distinction by which we called one as S and other as R strain? Now if R strain also developed a polysaccharide coat, then it will have all the properties of S strain, right? Now Griffith thought maybe something is transferred from the heat-killed S strain to R strain, because of which R strain is developing the polysaccharide coat around itself and it has become virulent. कुछ ना कुछ ऐसा था जो S strain, भले ही heat-killed S strain था, लेकिन उस S strain से R strain में कुछ ना कुछ ट्रांसफ़र हुआ, जिसकी वजह से R strain में भी polysaccharide coat डेवलप हो गया and it has become virulent. Now the question is, what is that getting transferred? Now, to trust his thought further, what did Griffith do? Even after the fourth experiment was done, Griffith tried to do some research on dead mice. Right? Confirmation तो करना था ना? So he tried to do some research on the dead mice, right? And he found that live S bacteria was recovered from the dead mice. Fourth case में dead mice से living S strain recover हुआ. Please remember, what did we inject in the dead mice? Live R strain और heat killed S strain. और क्या हमने recover किया? Live S strain. But now when the mice is dead and some is investigated was done in the body of mouse, it was found that live S bacteria was present. This is all confirmed his thought. यही सोचा था उन्होंने कि R strain S strain में convert हुई और उसके चारों ओर polysaccharide कोट डेवलप हो गया और वो virulent हो गई. And this must be due to the transfer of genetic material. There is some transforming principle which transferred or which transformed S strain from the R strain, right? तभी इसे नाम दिया गया transforming principle, right? Now the focus was to finding out the biochemical nature of transforming principle. अब देखना ये था कि बायोकेमिकल नेचर क्या है इस ट्रांसफॉर्मिंग प्रिंसिपल की, कैसे ट्रांसफॉर्म हो गया. सो नाउ रिगार्डिंग द बायोकेमिकल नेचर ऑफ ट्रांसफॉर्मिंग प्रिंसिपल, सो ट्रांसफॉर्मिंग प्रिंसिपल वाज द नेम गिवन टू द सब्सटेंस विच इज़ गेटिंग पास्ड ऑन फ्रॉम वन बैक्टीरिया टू द अदर बैक्टीरिया. वो सिर्फ उस सब्सटेंस को उन्होंने नाम दिया ट्रांसफॉर्मिंग प्रिंसिपल. सो दैट सब्सटेंस वर कॉल्ड एज ट्रांसफॉर्मिंग प्रिंसिपल बाय ग्रिफिथ. In order to know the biochemical nature of transforming principle, somebody thought that it could be a protein, right? Some of them thought it could also be a fat or carbohydrate, or it could be a nucleic acids. So there were different opinions then what it could be. Bacteriologists perform a series of experiments to identify the transforming principle. So some of the experiments which they perform were, like the very first thing, the precipitate of transforming principle with alcohol, and it was found that it was not a carbohydrate. अगर ये कार्बोहाइड्रेट नहीं होता तो एल्कोहल के साथ प्रेसिपिटेट नहीं होता, कोई प्रेसिपिटेट नहीं देता. But it precipitated with alcohol, so that is why it is not a carbohydrate. Okay, so these are some of the properties of different biomolecules, like properties of carbohydrate, protein and all. I hope you are aware of them. So they also found that transforming principle could not be destroyed with proteases. Proteases are the enzyme which digests the protein. So if you mix protein with proteases, it will destroy them, right? अगर protease इन्हें digest कर देता तो वो destroy हो जाते. अब ये destroy नहीं हुए. That means transforming principle is not a protein. It was also found that transforming principle could not be destroyed by lipase. ये भी देखा गया कि वो lipase से भी digest नहीं हुए. That means they were neither protein nor lipid और ना ही वो carbohydrate है. So what are they? So these transforming principles are like little similar with the nucleic acids. Whatever they might be rich in nucleic acid, but again it is a nucleic acid. Now children, we know about the two type of nucleic acid, RNA and DNA. So what could be that? या तो ये RNA होगा या ये DNA होगा. Then it was tried with enzyme which digest RNA, that is ribonuclease. इसे ribonuclease के साथ try किया. So he observed that transforming principle could not be inactivated with ribonucleases. So what did this prove? This proved that it was not an RNA. Now the only option which was left is the DNA, right? So they found when they use deoxyribose nuclease enzyme, DNase, then they observed that transforming principle could be inactivated, and it proving that it was a DNA. So when the DNA digesting enzyme were added to it, transforming principle inactivated, so it was a DNA. So it proves as a DNA. This is how he observed that genetic material, or the material which passed on from one generation to the next generation, it is nothing but it is a DNA. Now it is proved. DNA is the genetic material. So this was finally proved after Griffith Experiment, when Griffith defined transforming principle, and finally the biochemical nature of transforming principle notes as DNA. Okay, so by that it is proved that it is a DNA, right? Now one question is there, that can you think of any difference between DNAs and DNAs? इन दोनों में आपको क्या डिफरेंस समझ में आता है? This is the question for you as a homework. Write the answer in your WhatsApp group, biology WhatsApp group, and send to me, right? Now I hope that you understand the concept of genetic material, that how it was searched that DNA is the genetic material. I hope that you understand the Griffith experiment. Children, please do revise whatever we have studied today. So in the next video we will continue with further explanation. Till then, have a nice day and thank you.